Good morning, everyone, and thanks for letting me talk here. So, I talk a little bit. Yeah. Why don't you make a talk and send me a title? So I sent him a title, and then I had an email back. Look, this is your title. So here's Jim's title. So I was my youngest daughter when we did an art project. She said, I don't know what to do. So I racked my brain to come up with an idea. Immediately when I said that, she had another idea. And then I learned later I can say anything, she comes up with another idea. Dynamics of neurons in culture. So there's a big switch to the talks that we heard before. We're actually moving up in the animal kingdom. So we heard about the use of Drosophila, the use of C. elegans, invertebrates, great advantages because you can just put them on a microscope and look right through it, right? Well, if you go up in the animal kingdom to mice, rats, humans, that's impossible. You know, we have some techniques now. You can cut a little hole in the skull of the mouse. You can look at neurons right at the very top of the cortex, but that's about it. So if you want to understand anything more in detail about the function and structure of the nervous system of vertebrates, you have to take that nervous tissue out of the animal. Right? And that leads us to various forms of culture technologies. I mentioned a few, show you some of the data, that how you can acquire data, and kind of this new way to culture. I don't talk too much about it, but I'll just let you know that we go from 2 dimensions, so to speak, to 3 dimensions, and what that really means for the neuroscience. So, pretty much the very moment you take tissue out of the animal into culture, you do a sort of a reductionist experimental approach. You try to mimic different aspects of neurodevelopment, maintenance of neural connectivity in the adult animal, synaptic plasticity, regeneration events, and also degeneration events. The culture systems we use, they can be dissociated cells, explants, so you literally just rip a piece of tissue out and put it in a dish and hope it survives. You can make tissue slices, and then a little bit newer is a 3-dimensional way of culturing cells. When you culture the cells, in essence, you use a particular condition. We call this a basal medium. It's like a Gatorade. Glucose, sodium, calcium, vitamins, amino acids, whatever is really important for metabolism. Then you have additives or supplements that could be specific growth factors, other goodies. Sometimes we don't know those in particular. We can use feeder layers, so we use other cells that secrete all kind of molecules that are extremely supportive for the growth and survival of neurons. A simple, relatively cheap source of growth factors is serum. You know, we use fetal bovine serum. It's readily available. It's also fetal source, which means there's generally more growth factors in there. Very often we use antibiotics, although we try everything, to work sterile. Adding some antibiotics is not a bad thing. So, talking about nervous tissue in vertebrates, you have to mention both of these individuals, Camillo Golgi, an Italian, and Santiago Ramon y Cajal, a Spaniard. They're really the great-grandfathers of neuroscience. Golgi, you remember the name. There is the Golgi apparatus in cells, and he developed a silver impregnation staining technique that was unprecedented in detail. Because in those days, that was the best microscope you had. It's called bright field, one objective, that's it. So Santiago actually used the technology that Golgi developed, made it better. And those are some of the hand drawings that Santiago did looking through this microscope. And the level of detail that this man, found and basically put down with a fountain pen is pretty amazing. This is a pyramidal neuron. This is a portion of a dendrite with dendritic boutons. Here, a section through the cortex. And it was really those two individuals, in particular Santiago, that were able to explain that the nervous system is not a reticulum, a continuous system of tubules through your body, but it's made of individual cells. It was also Santiago, through these stainings, that showed that neurons make a projection and grow, and they call it a growth cone at the tip. So the amount of information is, we should have respect. He also got a Nobel Prize for that. Then a century later, a couple of $100 million, you can still do the same thing. However, they are now not hand-drawn, so there's a little bit less subjectivity to it. But this is a pyramidal neuron, 3D reconstruction of a dendritic branch with all the spines, and here is a cut section for the cortex. So the nervous tissue of vertebrates is characterized by an immense amount of complexity on a morphological level. So here, this is a cut through the hippocampus. So it's this classic S-shaped form. You know, the hippocampus thing is the Latin or Greek word for the seahorse, which is shaped like that. This is a section through the cerebellum. These neurons here are Purkinje neurons with an enormous, elaborate dendritic arborization. And then here is a neuron culture stained for these spines. So the complexity goes from the whole tissue all the way down to an individual cell. And just because I mentioned these spines here, the next slide has not much to do with the talk, except I like it. The spines are plastic. And in neuroplasticity, that you see is literally a footprint of your lifestyle. So during a task of learning or memory formation, you can observe that was actually taken from a live mouse where you cut a hole in the skull, and they taught the mouse, I think, to jump over a little hurdle on a treadmill. And the mouse was learning this, and they could show how spines form, go away, get bigger, move a little bit away. So there's an enormous amount of plasticity. This here is another example. Animals that were under a very strong stress versus animals that were kept on a sort of a low but very chronic stress. And the spine morphology changes under chronic stress. So if you think about, a lot of people constantly being stressed, not like, when you just put way too much stuff in your timetable and you run around, but stress like you don't know how to put food on the table, how to pay the rent, and those kind of things. It actually causes a slight degeneration of the spines. The individual that drank too much alcohol, the spines get really big, they kind of balloon up. Spines in a young individual, about 20 years old individual at 80. This person doesn't have any cognitive decline, but you can tell there's a little bit, no, that's not. That is from my brain. But even though in this older person there are less spines, there's no cognitive decline. When it comes to a person with Alzheimer's, the decline of the spines is even more dramatic. This is an interesting one. This was a rat addicted to cocaine, massive amount increase of spine formation. The idea is that the animal makes way more synapses and therefore starts to get this drive to find this reward, which is in essence addiction. And this is the latest, again, a rat controlled high fructose corn syrup diet, changes in spines. So, and what applies to rats applies to us too. So spines really leave a footprint of your lifestyle. In the vertebrate nervous system, we just heard C. elegans had 302 neurons, right? And they have to connect them now, so they know all the connections. You can probably forget this in a higher vertebrate. There are a lot of cells, a lot of connections. This is a reconstruction from an electron micrograph. You see the enormous density of neurons. and connections. This is an EM section, so here's the axon hillock, here's the axon. There is not a lot of empty space. This is packed. And this is the main connections in the human brain from the top, from the bottom. So here's the corpus callosum, left and right hemisphere. So there's a lot going on in our brains. And sometimes. Yeah. And so we're taking now these structures and trying to put them in model system to study events in development, maintenance, and regeneration. And so just to give you, you'll remember that picture of the hippocampus. This is another slice. This is in a brain slice in culture. So you see a neuron, here is an astrocyte. That's all great, but here are activated microglia cells. Microglia cells is part of the innate immune system that's in the brain. They respond very rapidly to insults. You don't want to have activated microglia in your brain because then you have a big brain inflammation, and that's not a good thing. So we have now a system that maintains a lot of the structure of the nervous system, but it sort of parallels with the brain injury because these macrophages, or the microglia, they're activated. And they send or secrete a lot of cytotoxic substances out. So this is a dissociated culture, long-term culture of hippocampal neurons. So this morphology and structure versus this morphology and structure, you don't have to go to school. You realize that's not the same. And then we even make some cell lines for hippocampal neurons, which don't look any more like neurons at all. Another example down here, this is, you saw that picture before, this is a cross-section to the cerebellum. Purkinje neurons, this is a slice in culture. This is the Purkinje cell layer right here, a little bit less pronounced. And this is a Purkinje neuron in culture. There is something reminiscent, but you start losing information, obviously. So when you go into the neuronal tissue culture, you will lose complexity. Do this on purpose in order to be able to focus on aspects of the nervous system, but it could also introduce mistakes. You lose a lot of morphological detail. The contacts among cells and their density drops dramatically, especially when you think about going into the associated cell culture. And also, most neuronal tissue cultures, if not all, of higher vertebrates rely on embryonic tissue. Rat EAT, cortical neurons. Let's study Alzheimer's. You know, people when they're 2 years old don't have Alzheimer's. We did literally test over 1,000 drugs against stroke in rats, which in human years are about comparable to a 20-year-old human. But most people don't have a stroke at 20. They are 60 or 70. So, and it was actually at a meeting, and we asked the speaker, why don't you take old rats? The answer is simple, too expensive. You can't take a rat and keep it in a cage for 3 years or 2.5 before you make an experiment. So we also use genetically homogeneous animals that gives us a better signal-to-noise ratio when we make our analysis. But when we take that data and go out into this room, it kind of fails because we are not genetically homogeneous. In your culture conditions, you know, sometimes we don't think about that. We use often kind of peculiar conditions. I just point out a couple. Tissue culture, you know, it's an incubator. You put room air in the back, 5% CO2. Room air has about, you know, 18 to 20% oxygen. It's 160 millimeter mercury, sorry for the mistake. That's the partial oxygen pressure. In blood, it's really only in your own blood, whole physiology, it's 80 millimeter. In the brain, about 25 to 50. So we're having neurons in a cell culture incubator that have 8 times more partial oxygen than in your brain. And I do this. And on top of that, I study oxidative stress. And you wonder, how oxidatively stressed are my neurons already before I actually stress them. So glucose is another great example. In the blood, 3 to 7 millimolar. If you go above 7 millimolar, you're not going to feel great. And it becomes actually quite dangerous. In the brain, newer data, somewhere around 1 to 3 micromolar. Our basal media, like EMEM or neurobasal, is 25 to 50 millimolar glucose. So if we do cultured neurons experiments, we have somebody that has a severe hyperglycemic shock combined with a massive oxidative stress, and that is our basal condition. That's our control. So, you wonder how compromised our results are. We also use particular substrates, like plastic, which you usually don't find in your brain. Poly-D-lysine, the amino acids are toxic, but poly-D-lysine is actually a great substrate, very sticky. We use, endogenous molecules like laminin or fibronectin or collagen. And now we're also moving towards like a mixture of, we call it extracellular matrix, like gels or molecules or mixtures, matrices. So we have to really think about how our reductionist approach could actually compromise the data. So the dynamics of neurons persist throughout lifespan, starts in development, and really never stops until we die. It's just different portions of neurons that show motility. The motility is absolutely vital, obviously, to develop the nervous system, to maintain and to regenerate connectivity. Adhesion, contacts to substrates or cells has a great influence on this motility and the connectivity. But it is interesting that most or a large part of this dynamic or motility is tied to a very well-known molecule, actin, and its ability to form filaments and depolarize and repolymerize in conjunction with myosin family members in order to generate force and obviously tubulin. That's pretty much the essential network, the cytoskeleton, and that's very dynamic and responds to outside influences. So this is the picture of the end of a growing neurite. So here's the neurite cell body, will be somewhere down here. You have this structure that has this lot like big veils and spikes. They're called phylopodia. This is a growth cone. This is typical for an elongating neural process, very motile, very, very fast. Lots of sensory capacity for many, many different signals, to some degree almost autonomous. In the Cater lab, we actually cut a growth cone right off the neuron, and it's still responding to acetylcholine and they turn. Didn't need the cell body, knew it by, just like that. And it's this actin-based motility, polymerization at the periphery, depolarization in the back, and it drives protrusion of membranes. Growth cones, they're very busy. They constantly take information in. This is attractive or repulsive information. So attractive information can be soluble molecules in a gradient or adhesive molecules. The growth cone turns towards the molecules, or the growth cone is repelled from molecules. This is a number of molecules that all kind of repel growth cones and don't like them. In essence, these signals are translated through membrane receptors and some Rho GTPase family members into a change in the actin cytoskeleton, and then you see in the microscope change in behavior, turning, slowing down, speeding up. So, too quick. This is so these are neural processes. Here are some growth cones. The cell bodies are hidden, and it's just a short video that shows you the motility. And you see how they move forward. Right? That's obviously sped up. So that's on the control condition, hypoglycemic oxidative stress, to dissociate the neurite culture. Here, the same kind of neurons are the spinal cord neurons, but they are going to see a pro-inflammatory cytokine shortly after the movie. If I can started. So they're doing okay, cytokines put on, and they obviously don't like it. This is a cytokine that can show up in your brain within 10 minutes after you get a really hard hit on your head. Your neuronal plasticity does not like that. We actually went and studied this in a lot more detail because of its relation to aging and neurodegeneration, where oxidative stress and inflammation is a major driving force of the degeneration. So if you follow these neurons, the tips of those neurons in a control condition over time, you can just look at the displacement of the growth cone over time, and it kind of follows a straight line. If we add a lot of TNF-alpha, the pro-inflammatory cytokine, they'll stop very quickly, they stall, and actually retract, and they stop growth. They're just dead. It can't move on. The motility mechanism is shut down. Down here, these two lines is the same thing, this TNF-alpha, but we try to scavenge oxygen radicals, and we try to block the NADPH oxidase, and we can rescue quite an amount of the motility machinery. Not fully, but quite a bit. So the idea came up that cytokines elicit an oxidative stress, and it just freezes up the cytoskeleton. This is just microscopy, so that's about 3 minutes before the growth cone sees an oxygen radical for TNF-alpha, and then the oxygen radical concentration goes way up in the growth cone, stays high, and the growth cone seizes its motility. If we do this with the cell line, what we find is that when we add TNF-alpha, the cells actually respond first positive. They make a lot of protrusion veils. They get very active for a certain amount of time, and then the actin cytoskeleton just freezes up. And we found a huge amount of carbonylation of actin in those neurons. So basically, our conclusion was the actin gets oxidized and is not functional anymore. Then the neural motility dies. This was actually done when I was a postdoc in Jim's lab. So we looked at these GGPases, and it kind of started with our later work putting together the puzzle that we have pro-inflammatory cytokines. They signal through the RAC1A protein and generate ROS signals, causes a reorganization of the actin, and it changes growth cone motility. And this is sort of shows it a little better. The idea that for everything in biology, there is an optimal range. Too much or too little is detrimental. So if we have too little RAC activity, our growth cone sees their motility. If we exaggerated RAC activity, they also sees motility. They didn't like it. They just want to have the right amount of actin and RAC activity. And this change in RAC activity correlated with an increase in oxygen radicals. So if we have optimal conditions, we increase the oxygen radical pressure, the motility degenerates. Ironically, if we load the neurons up with a massive amount of antioxidants, they didn't like that either. They actually stopped growing. So which basically told us growth cones want to have a little bit of oxygen radicals in order to actually function. So oxygen radicals are not just bad. There is a physiological range and there's a pathological range of the concentration. Another problem that we tried to tackle in cell culture was if you think about an injury in the nervous system, like a spinal cord injury, and you look at the actual morphology or anatomy of the neurons, you have neurons like motor neurons that are up in your motor cortex and project all the way down. Other motor neurons are in your spinal cord. So if you have an injury, some motor neurons see that pro-inflammatory signal at the cell body or the dendrites, and other neurons see like way out on the axon. And the question is that the same signal? Does that generate the same response in the neurons? So to get this into the cell culture, we decided that we're going to immobilize the signal on little beads and then replace the beads to different portions of the neuron. And we used the laser tweezer for that. It's basically a laser beam where you can just trap the bead, and then you can move your culture dish around and you can literally decide where you want to place the bead. So in this case, I placed the bead a little bit in front of the neuron. The motor neuron approaches a bead that's coated with TNF-alpha, and it goes through a bunch of, touch and release kind of pharmacopoeia. It attaches, then it starts to make some weird level of podium formation, and ultimately it just degenerates. It just freezes right up on the bead. If I take the same bead and boil it in hot water, so I kill the TNF-alpha on the bead, the growth cone doesn't care. It just moves right by. So we did all kind of experiments like that. In the Taylor lab, this was a little bit more based on the newer research that just came out in grasshopper embryos that showed these guidepost cells, so that neurons that are in the periphery send out a process, and the process is guided into the central nervous system. And at very particular spots in the embryo, the growth cone makes a sharp turn. And it had to do with one particular cell. They went in with a laser to ablate the cell, the growth cone completely went off the track. And so this was a great analogy for our beads. So I had neurons on polylysine, but they kind of grow a little bit on the slow side, and then I popped the laminin-coated bead. Laminin is a great substrate for neurons. Put it right in front of the neuron that I knew it's going to find with the photopodia. Then the growth cone made contact, was a little bit around the bead, and then it just sped up. So they were around 30 micrometers per hour growth speed before, a little bit of stalling, and then it took off at about 80 micrometer per hour for a certain amount of time past the bead, and then the growth slowed down. It's almost like you could just give this growth cone a signal, and it sort of ignored it wasn't polylysine, and the machinery just got kicked in like it's on laminin, but it wasn't long lasting. If I did the same experiment, but I just plugged the bead right onto the neurite, the growth never changed. Because the neurite doesn't have the reception to understand and drive the motility machinery. It's really on the growth cone. And then this was more of a game. You just, you play around in the lab literally, and it just works. So I put a bead here, pulled the growth cone here, went up here, had those conformation here, B2, 3, and 4. But the growth cone decided to go over the 4. I have no idea why. Maybe we had more laminin. And then another bead, 5. And so if I place the beads in the right distance, the growth cone kept growing like they were a laminin. They completely ignored the fact that they were really actually stuck or polymized. So, and that led basically us to then look at signaling pathways of how the growth cones actually read a molecule in the environment, translate this into the decision of how motile the growth cone decides to be. So when it comes to neurodegeneration, the motility has something to do with connectivity. So there are two large camps in the neurodegeneration field. The people that say you have to lose neurons and then your brain doesn't work anymore. And then the others, like me or like Jim, they say you can keep all the neurons in the brain. If they're not connected, nothing works. And so I'm a connected guy. So if you lose the connectivity, the integrity and plasticity, you lose function. So neurodegeneration, really early on, we should look at synaptic events. And oxidative stress is important in that. It's a little bit our working model. It is interesting that a lot of these sort of toxic signals or neurodegenerative signals seem to be co-localized in lipid rafts, these little microdomains in plasma membranes that are rich in sphingolipid, cholesterol, and phosphinophores. And so we know now that different molecules, the pro-inflammatory cytokines, the amyloid beta, and one of my graduate students showed that the coprotein of the HIV virus, which also causes neurodegenerative problems, so cognitive problems in AIDS patients that grow older, they all seem to funnel through the same pathway that involves the prion protein, the NADPH oxidase generating oxidative radicals, and leading to these actin rod formation, these rod-like inclusions that seem to just suck not the oxygen but the actin out of the neurite, and just then you lose synaptic activity, you lose motility, and that seems to lead to degeneration. So, 3D cultures. Why do we move to 3D cultures? Because this is a neuron in a 2D culture, and this is a neuron in a 3D culture. And this is a reconstructed neuron out of an EM page. So that looks a lot more like that. And so when we have neurons in basically 2 dimensions, they don't have the cell shape the way they usually have in vivo. They also have different ways of how they form their neurites and how they make connectivity. So we have now learned that it might be important to give the nervous tissue a much more in vivo-like surrounding in order to study what is actually going on. Which obviously makes you think like, wow, did we do something really wrong for the last 50 years? It is possible. But you know, it's better to learn something at one point than just keep ignoring it. So these 3D scaffolds provide way more adhesive contacts, a higher cell density, a higher density among the processes. So more proximity to neighboring cells, they reduce intracellular space, because if you think of this, if you have a culture dish, you know, a 60-millimeter culture dish with neurons growing on the bottom, and that pipette in TNF-alpha, it takes a while for this to kind of spread out. This is not really a scenario that you find in vivo. And so it is possible that with this higher density in vivo, that's for some molecules, it's not so easy to quickly diffuse into the neural tissue, and the neurons might actually respond differently. And so on this table, they kind of listed a little bit of differences between 2 and 3D cultures. So in neurons, in 2D cultures are mostly sheet-like and flat in a monolayer. In the 3D, they are more in a spheroid aggregate, round cell body state, which is more natural. They talk about quite dramatic changes in proliferation. Cells respond different to drugs. They have done studies with platinum compounds in cancer cells in 3D cultures. The cells don't respond very well to the platinum compound. You put them in 2D cultures, you kill them. Right? So all our drug discovery, the pharmaceutical industry, are cell lines grown in a monolayer. And that's how we drive our drug discovery. So you start to wonder, have we missed a bunch of things because we used the wrong culture system? Cell cycle is a little bit different. And then this, you know, is something that we worry. They have shown that not just a few, but a whole slew of genes are very differently expressed, whether your cells are in this 3D situation versus 2D. Not that I have read up or seen anything, but I'm pretty sure that probably the epigenetic makeup of the cells might also change depending on 3D versus 2D situation. So this is a little bit similar that I just put together from other papers. Why did we go for 2D cultures? Well, sometimes we do things in science because it works. And then you go on, you know, you can manipulate neurons. It's a lot easier in a culture dish to go and put a patch pipette on the neuron than going first 4 millimeters through the cortex to this particular cell, right? So obviously, but you know, there's also an enormous amount of literature, which is nice. But we do know there is way too much surface exposure to the environment and what neurons really have in vivo. So in the 3D cultures, we have the higher density, slower diffusion, probably more real gradients of molecules. They did a couple of tests with gamma secretase and the BACE inhibitor that has to do with Alzheimer's. And in 2D cultures, these inhibitors were very potent in rescuing the neurons from A-beta toxicity. In 3D cultures, they didn't really work almost at all. So this is probably the future, you know, of cell culture, not just neuroscience, to really try to mimic better the actual nervous tissue situation. You know, the ways, so what are the lessons that we have learned? Neurons in 3D respond quite different to signals or pharmacology or, you know, in vivo signals. Neuron outgrowth is actually enhanced and it's different in the way they're sprouting. Synaptic density is actually greatly increased. Ion channel physiology, probably someone can talk more about that, is dramatically different. whether the neuron is a 2D versus a 3D situation, and then reports about very low gene expression changes between the two different kind of culture systems. So yeah, I just quickly want to mention a few people that helped me do all this. So some graduate students over here, Lisa in particular, undergraduate students that have been involved over the years, some of my collaborators, and a couple of research professionals in the lab that made everything work while I go and talk. Thank you very much.
The cells to make rod when you subject it to the oxygen species and all. Do you feel like there's a difference in the composition, the receptor composition, the lipid wrap between these different cell types, and does it change in gene development?
You know, I can only speculate on that. But
Who's
going to find out in the next 6 months?
I hope it's 6 months. But you know, it is really very possible that if you have the cells in a 3D system and it affects gene expression, that I mean, it would be really interesting to see whether the lipid wrap composition in the 3D culture is different than in the 2D system.
No. Between the 20% cells that are actually making rods and the 20% and the 80% cells that are not making rods, I think the number is pretty much the same between stable 3D models and in culture model too. That's what my knowledge was. But does that?
So if you take hippocampal neurons, right, you take the whole hippocampus out of the CA1 all the way to the CA3, right, they dissociate that. And we don't know whether CA1 neurons are exactly like a CA2 neuron. And maybe some of them are more prone for the rod formation. So a little bit dissociated from that, we did studies with cerebellar neurons versus cortical.
Some way it's possible to downregulate those receptors and actually get rid of the rod formation in the cells in terms of potential therapy. That's what I was.
Well, maybe, yeah. Well, so we are actually trying to go in and kind of fiddle around with the NADPH oxidating neurons. And when we do that, just like with prion null mice, you don't get the rod formation. So those are gatekeepers. You kind of manipulate the prion or the NADPH oxidase and you don't get the rod formation downstream of those signals. The problem with the NADPH oxidase, nobody has really ever. We know the protein for 40 years, and we have just the poorest pharmacology whatsoever. The most used molecule is called diphenylalanium, or DPI, which I use. Chemically, it inhibits any process in a cell that requires a 1-electron transport. That's about as specific as we get. You know, but we do know that if you manipulate certain isoforms of the NADPH oxidase, that the nervous tissue is much more resistant to certain insults.
I can add sort of one additional thing in terms of signaling for rod formation. There seems to be, at least in one pathway for rod formation, it's dependent on a specific cyclic AMP phosphodiesterase. And there are like 25 isoforms of this that are made in brain, and there are particular subpopulations of neurons in the hippocampus that express one particular isoform that seems to be in neurons whose spines disappear in response to certain types of stress, and that could be a population that is really responding with rods. It may not be a sort of general phenomenon of every cell, but it may also depend on many other factors that we don't know.